Welcome to this session entitled A Clinical Trial and Research Database Derived from Electronic Patient Records. My name is Ian Green. I'm the Customer Relations Lead for Europe and Clinical Engagement Business Manager for SNOMED International, and I'll be moderating this session, and the presenter is Sophie Williams. So Sophie, over to you.
Thank you very much. So I'm Sophie Williams. I'm a research data scientist at Barts Health NHS Trust, and I'm going to be telling you a bit about our work going on in the Trust, developing a COVID-19 clinical trial and research database, which is derived from electronic patient records. And in this talk, I'm really going to try and demonstrate some of the practical uses of SNOMED CT that we've done for clinical data analytics. So just to give you a brief outline of what I'll be talking about. So first I'll tell you a bit about how we collect data and its uses. Then I'll give you a bit of background about the COVID-19 pandemic situation in East London to date. I'll then tell you a bit about our work in text analytics to enhance the data set, and then how we've been using SNOMED expression constraint language for clinical analytics. And we've kind of been doing a few different things in this bracket. So we've been using ECL for defining and categorizing concepts within the data set. We've been using ECL for some statistical analysis and also for some predictive modeling. And finally, I'm going to be giving you a bit of an overview about some of the results coming out of our COVID data set. So just to tell you about where our data comes from, really. Within Barts Health, we use Cerner Millennium, and we're one of the largest NHS trusts, so within the UK. Barts Health covers 5 hospitals, and throughout the pandemic, we also were the lead operational one field hospital, which was not open for very long. And in usual times, we see 2.5 million patients per year, so it's a very large trust, and it covers a diverse East London population. So our Cerner Millennium Health Record, administrative and clinical information. It has a longitudinal record of over 10 million patients, which has been developed over a 12-year period. So this is a really longitudinal record that goes back, but obviously the contents and the amount of data within the record varies the longer you go back, the kind of more sparse it is. Also, the level of use varies across specialties, but there's a really big drive to become paper-free across the trust, and this is including at the NHS Nightingale London, which was the field hospital. So within our data warehouse, we have both these administrative commissioning data sets, which are sent out to kind of the national NHS bodies, but we also have the health professional completed data, which is entered into the EHR. The quality of data between patients obviously varies, because if a patient has a lot of encounters in the hospital, then they're likely to have a much fuller record than if they kind of come to Barts Health for the first time. There's also a lot of different uses of different ways of SNOMED being used in the data warehouse. Just to give you an example, so we have the emergency care data sets, which contain only a subset of SNOMED concept IDs, and they're used to record the chief complaint and the diagnosis when a patient enters ED. We store description IDs from SNOMED in our data warehouse, whereas the concept IDs are what we use in the analysis. And because we have such a longitudinal record, many codes that have been entered historically are now not active anymore, and so these are also not currently supported for analytics. So there's kind of a few tweaks we have to do before we get the data into a version that's kind of useful. So just to tell you a bit about the kind of background situation of COVID in East London. So within the trust, I think these are already a couple of weeks out of date. And we had 2,500 inpatients were recovered and were discharged we came in with COVID and we had 654 COVID-related deaths. The field hospital, NHS Nightingale, was stepped down after treating just 54 patients. And one of our kind of key things that happened during the dealing with the pandemic is we had an operational team that was set up to recruit patients into clinical trials. And we specifically looked at the kind of WHO and the NIHR priority clinical trials. And as part of this, there were obviously many requests to access our COVID-19 patient data, but it was not feasible to allow different people broad access to the entire data warehouse, as this is solely used for direct care and kind of running the hospital purposes. And so I was tasked with preparing a kind of standardized data set for all the different uses, including clinical trials, both to enroll patients onto the priority trials and to reduce manual data entry to report data back to the prioritized clinical trials, and also vaccine trials. We use this data set for internal audit. This is to better understand the population of COVID-19 patients at BARTS and to look into how outcomes are related to demographics, comorbidities, virus symptoms and treatments. And finally, research. So we work closely with our neighbouring university, Queen Mary's, and there are many statisticians and epidemiologists there who have research studies. So we shared this data set initially just with them, but we're keen to work with research consortiums now to kind of get wider external use of the data set to contribute to large studies. So just to go over where the data comes from, I kind of mentioned before that we have both administrative, healthcare professional input, and also automated devices that all feed data into our Cerner Millennium system. And then this is merged with other clinical systems across the trust. And our data warehouse team performs some magic on it to get it into a good structure in the data warehouse, which is a SQL server. And I directly extract my data sets from this data warehouse. I do processing on it and do some text analytics work. And the COVID data set contains things such as demographics, the encounters information, and really rich clinical content on comorbidities, symptoms, complications, lab test results, and vital signs. And there is free text available, which we really use to enhance the other fields using text processing. And then from this one kind of standardized data set, we can pull extracts out, which are filtered based on the requirements of different trials, or pseudonymized extracts for research purposes. So as you've probably got the impression, this is very much a cleaned but far from perfect or research grade data set. It contains structured data such as SNOMED terms, numerical data such as platelet count and strings in the discharge summary. But one of the really key things that we've found is very much needed is that we're having sanity checks by clinicians along the way. Just, do these values make sense? Should we include more things in the data set? And that's kind of been the only way that it's been a really true good picture of the situation. We've been performing linkages to other data sets as needed. So these kind of include OpenGov data and other local data sets that clinical teams might have their own data sets open collecting, recording kind of special things. You can see here that these figures just kind of give a bit of an overview of the picture. So we've got 3,000 confirmed patients and an additional 1,000 suspected COVID patients in the data set. There's more males than females. And although the population is slightly older, there are patients all the way from 20 to 100 years old. So it's really a very broad age range that's been attending the hospital. And as mentioned before, we have a really broad ethnic diversity of patients from East London. Now, I mentioned that we use a lot of the SNOMED coded data, and using just the coded data, we can pull out the most common previous medical history conditions. So hypertension is coming up, which has come up in the literature. But you'll also notice on this graph on the left that the majority of patients actually don't have any previous conditions recorded, which is kind of one of the things that comes up when you speak to the clinicians, that most of the patients do have a medical history, whether it's been recorded completely or not. And similarly with symptoms, you can see in the bottom graph that shortness of breath has been pretty well recorded, but again, most patients have 0 or 1 symptoms recorded, whereas we know that there's kind of multiple symptoms come up when a patient is diagnosed with coronavirus. So kind of seeing this as the initial, this is the level of richness of the coded data. We've been doing additional work using text processing to enhance the data. And this is because when you want to know where all the rich clinical information is, it's within the free text notes that nurses record that are included in the discharge summary or included in the admission notes. So, and this information is otherwise inaccessible if we don't extract it out with NLP. The benefit of extracting it out and matching to SNOMED codes, which is what we focused on, is that then the process data can be included in all the other analytics that we're doing. And also this data is anonymized, whereas free text notes are full of identifiers, so aren't really appropriate to be used for research. And it's just better for analysis. So we've been using open source NLP tools, mainly based around the Stanford NLP tools, to extract just the required information from clinical notes. So we've kind of scaled down our NLP task to just search for the fields that we're already collecting for this data set, which has made the task a bit easier. And so we've used bag of words to pull up the clinical terms, and then we've been matching to a restricted dictionary of SNOMED codes for the clinical information or BNS codes for medications. And then a really important step is that we've been identifying negated terms, so where it's recorded that patient did not have shortness of breath, and we've removed these from the final list. So here's an example of a X-ray chest report. So you can see underlined all the clinical terms that we might be interested in. And then through the NLP process, you can see this is the kind of way it looks once we call out the SNOMED terms. And we're seeing around a third enhancement in data completion. And we've achieved pretty high accuracy, so around 90% for like positively identifying concepts that we want to be extracting out. And we're finding that it has over 90% accuracy in not pulling out negated terms, which is really good. But there's plenty more work to be done within the text processing area. So we're doing some ongoing work with external collaborators now, because multiple different trusts, not even just in the UK, globally, we're all kind of working on the same problems here. So we're focused now on looking at qualifiers. So quite often someone will put like question mark pneumonia, and that's where it's definitely not confirmed, it's just a suspicion they have, or check for ARDS. So we want to be able to pull out the kind of qualifiers. Also, timing is really key in research. If a patient had a stroke 10 years ago, it's quite a different situation to if they had it last month. And quite often with symptoms, they record something like 4/7, which will be like 4 out of 4 days in the past week they've been coughing. And this is obviously rich clinical content, so we would be really keen to extract this out. So then what do we do with all this nicely coded data once we have it either directly from the data warehouse or we've extracted it using NLP? So for the COVID data set, we are categorizing all the clinical terms into 34 categories. And these are mainly those that are defined in ISARIC, one of the big data collection studies, which is a WHO priority study. But then we've also added in some local additions from the infectious disease team here at Barts, and also by speaking to academic researchers who have specific things they're really interested in. And to do these definitions, we're using the relationships within SNOMED's OWL ontology to define the coded data, and that's using expression constraint language. And we find that ECL is really it's really useful because the SNOMED ontology is a structured polyhierarchical form, so that's really handy for answering analytical questions. I use the Snowstorm API to access the terminology server, so I can query it using ECL and then find matches in our data set to be able to add a label for our analytics or feature modeling. An example of this is that if we wanted to find diseases that have a sudden onset and are of short duration that only affect the interstitial tissue of the lung, I would write this ECL. So you can see first I'm defining that I'm only interested in SNOMED terms that are diseases, and then I've defined two relationships. So the first is that the clinical course must be sudden onset and or short duration, and the finding site must be a structure of interstitial tissue of lung. But we found that we kind of are often tweaking how we have defined our ECL queries to make sure we're adding in this specific condition as well. So there's a bit of a trial and error to writing them. And Onto Server by CSIRO has a really good ECL query builder, which I recommend for kind of designing your own ECL queries. However, within our data warehouse, I kind of mentioned that not all the clinical information is in a SNOMED CT form. We submit ICD-10 codes and APCS4 codes as part of our commissioning data sets. And although there is SNOMED CT to ICD-10 mapping, there's not vice versa. So currently we have to process these data sets in different ways, like separately, and then combine the results, which is obviously duplicating work, so not ideal. Additionally, for in the trust, we can use Power Forms, which is a Millennium tool for structured data collection. These are basically forms that are filled out, and the clinical teams design their own questions. And it's been especially useful over COVID, where there's kind of information that's been recorded that isn't usually recorded, such as kind of the ventilator type that a patient's on, or whether they've been traveling recently. And this kind of information is really, really key for our COVID data set. So we have to find ways to incorporate this into the analysis as well. So just to go over the ECL again, just to kind of explain how it's been useful in this data set. So if I defined that I was only interested in SNOMED concepts that were diseases, you can see that there are, or when I first wrote the slide, there were 76,000 or over 76,000 concept IDs that fitted that requirement. And then if I define down that I'm only interested in diseases with a clinical course that is a sudden onset and or short duration, then there's 1,530 terms that fit that requirement. Whereas if I am interested in any disease which has the finding site of a structure of interstitial tissue of the lung, then there's only 59 concepts that fit with that definition. And if I say I'm interested in things that have both of these relationships, then there are 9 concept IDs that fit this. And you can see here, this is one of the pages from the terminology browser, the acute respiratory distress syndrome is a very bad complication of coronavirus. You can see here that it's defined as it is a disease, and it has the relationship, the finding site relationship that I defined in ECL, and the clinical course relationship, and it has additional relationships that aren't relevant to my ECL query, but it doesn't matter. And different relationships will be relevant to different queries. So sometimes I'm interested in defining the finding site, and other times I'm not. This is just giving you some example extracts. So the top table shows an example of a problems list from our data warehouse, which has been completed by the clinicians entering SNOMED coded data into the Cerner Millennium system. So you can see there's patient identifier, encounter identifier, kind of free text wording of the problem, which is linked to the problem code, which is a SNOMED description ID. We also have the date of onset and a kind of qualifier, so the confirmed, suspected, eliminated, things like that. But because there's so many different problem codes that could be entered, we realized that it wouldn't be that useful just giving this to the researchers. They really want a nice clear table like the process tables that we've developed. So the middle table here is an example of how we've processed previous medical history, where it's just kind of a binary, has the patient had this condition in their past, yes or no? So you can see here there's cardiovascular disease, hypertension, asthma, and you can see linked to the pseudo-pseudonymization key, whether the patient has it or not. And then as coronavirus is an acute condition, it's only kind of been going on this year. For our presenting illness and complications data, we provided the date that there was first this condition or this complication or presenting symptom was first recorded either in the free text or in the structured data. So for each individual, they'll get the date or a null if they have no recorded information about that complication. And the feedback is that these tables are very nice, very easy to use. You just kind of stick them into your models. But what else have we done with clinical analytics and SNOMED data? So another project I'm working on is around diabetic foot disease, and we're using purely the SNOMED data within the data warehouse to see if we can extract features that we can use to predict which patients will go on to get vascular complications, including foot disease. And in this project, I'm using a pseudonymized data set of diabetes patients, and this is a cohort of 30,000 patients, and we can get SNOMED data from their diagnoses, problems lists, but also procedures, and then we link that with their encounters information and some basic demographics. So within just these 30,000 patients, there's 25,000 unique SNOMED codes. So without using UCL, there'd be absolutely no way of processing that volume of kind of diversity of how disorders are coded up. Whereas using UCL, the health professionals can use whichever code they think fits the diagnosis best, and then we can adapt to include that in the categories as we see fit. And in this data set, there's an average of 12 hospital encounters per person. So they've generally got pretty complete and well-coded information. So you can see in this diagram on the right, we are comparing different categories of diagnosis to the onset of a diabetic foot diagnosis. So these are all patients who do have diabetic foot. You can see around the diabetic complication, there's a peak. That is their diagnosis of diabetic foot. But then also interestingly, you can see there's quite a lot of recording of infectious diseases around the diabetic foot diagnosis. And there's also a lot of peaks around cardiovascular problems. Unfortunately, so the graph on the right is where they've been coded up without a date. And you can see a lot of the diabetes diagnoses, there is no date. So that's not so good for timing-based and predictive modeling, because you really need to know when the patient was diagnosed with the condition. Another piece of work we've been doing is using SNOMED International's health data analytics tool. So we've been using the statistical test API, which has enabled us to generate hazard ratios, again using the SNOMED CT coded dataset. So for this, we use ECL to define the patient cohort, which is diabetes patients, and to compare the incidence of an outcome with different exposures. So for this task, we've compared 2 risk factors towards the outcome of a lower limb amputation, which is quite often a complication of diabetic foot. And we've compared vascular disease and neuropathy. And you can see here that we've got a hazard ratio of over 2 for neuropathy. So patients who have a neuropathy diagnosis are over 2 times more likely to require a lower limb amputation than those without neuropathy. And for vascular disease, this is over 10. And these values fit with the literature, which is really exciting because it's showing that our real-world data can be used to perform analytics, which kind of makes sense, which is really nice. But I also said I would tell you a bit about how people have been using our COVID data set. So within the Trust, we've set up a data access board to both approve and prioritize studies. And this is in addition to the General Research Office, which approves clinical studies. So we're also intending to join research consortiums to allow wider use of the data set now that it's kind of in a consistent place. So within the Trust, there's already over 10 different research and audit studies using the data set. Some of the things they're doing are looking at outcomes by clinical cohort. So you can see in the top left diagram, they're comparing onset symptoms to final outcome, and also doing survival analysis based either on onset symptoms or on demographics. Also something we've started doing recently is validating external models that have been published in the literature. And this is so we can very much, as soon as kind of we know that a model works in our population, we can start using it in clinical settings straight away. And this work is all supported by the statisticians at Queen Mary University London. So I just wanted to conclude. So hopefully I've been showing in these slides that our electronic patient data is rich enough to share for data collection clinical trials. But this is only really possible with enhancement by text processing, especially for the kind of symptoms and complications data. The de-identified version of this data set is a valuable resource for research, but we've been required to develop data access rules, prioritization, and also a hosting platform for external access. So the SNOMED coded data can easily be categorized using ECL, but also ECL can be used using Snowstorm and form descriptive statistics on our data set. And just to finally say, this is definitely not just been my work. There's been a big group of collaborators, both within the Trust and externally, so they're all mentioned here. Thank you, and I'll look forward to hearing your questions.
So I'd like to thank Sophie for her presentations, and we'll now move from the recorded presentation over to a live Q&A. Thank you. Welcome to this live Q&A session. My name's Ian Green from SNOMED International. First of all, I'd like to thank Sophie for an excellent presentation. We have a number of questions we've been accumulating while you've been presenting. So where to start? What do you think we can do to improve data quality and interest in data entry by health professionals?
I think we found within the Trust that definitely getting the clinicians involved in the analysis, the results, and using the data really gives them a lot of incentive to make sure that the data being put into the data set is high quality, so that the results they get out they can actually use for their own interest and their own research. So we're trying to use more tools to get the different teams kind of getting to grips with the data set themselves and performing their own analytics. And I think, yeah, as soon as they're interested in and knowing how to use the data, then that's, yeah, so much incentive to improve the quality. And
what about the range of specialists and researchers that have made use of the core COVID data set so far?
Yeah, there is a really big range. So there's kind of clinical teams who have their own consented patient tissue banks, and they want to see if any of those patient cohorts have had COVID and link up with those data sets. There's clinical teams who want to do audits because they're for their own treatment of patients, ranging across lots of different specialties. And then there's these big national and global trials where we're kind of collecting data and sharing for them, as well as the clinical trials, which I go through. So yeah, it is a really broad range, which is really great.
Then a couple of technical questions for you. So what data model do you use to expose the data for further analysis? Is there anything linked to either OMOP or Odyssey in the way you work?
Yeah, so I've been following all the Odyssey work with loads of interest. At the moment, because this was very much a response to a pandemic, our data is just based on the structure of the data warehouse, which is broadly based on Cerner Millennium Systems, but with some changes. I think kind of developing this further in kind of longer term will be really good to get it into the OMOP Odyssey formats, but it's just not currently like that.
So you mentioned you undertake analysis using SNOMED concept IDs. Are you able to undertake analysis based on the attribute value relationships?
Yeah, so some of the definitions we do use the relationship attributes. It really depends on how we want to define the kind of groupings that we do. So some of them, it is fine just to kind of use the kind of more hierarchical structure, so kind of children of a term, whereas others, we want to be really specific based on either timing windows or kind of which body structure is affected. So it really depends on how we want to use the SNOMED concepts.
And how do you distinguish between patients' past history and the current ongoing conditions for your analysis? Do you use the record heading, yeah, or how do you?
So in our system, you can record the date of onset, which when it's recorded is really useful, and you just use that as the kind of cutoff. If that date was before they were admitted for their COVID encounter, then we're including it in previous medical history. And if its onset is after this encounter, then it's included in current. But there's many times when the date of onset is left blank, and then it kind of defaults to a ridiculous date, which makes no sense. So then we kind of have to decide whether to include it. And generally for past medical history, I just include that, whatever the date, because we kind of want to capture as much of the clinical data as possible.
Okay, and one final question. How do you expose the data for further analysis with other collaborators, or do you, is probably the first question. If you do, how do you do it?
So when we're just sharing the data internally, it's just kind of in a de-identified format, flat file structures that are shared, and we share it just over kind of secure internal networks. We're talking to HDR UK, which is one of the big medical data research consortiums across the UK, about how we can kind of get it onto a platform for sharing externally and sharing more broadly. But we're completely relying on there being kind of set up trusted research environments to enable us to do that. We're not doing that in-house at the moment.
Okay, so we've slightly overrun, so I'm gonna call an end to the questions. If there are further questions, or if there are questions we haven't answered, or further questions, I'm sure Sophie would be happy to answer them via email.
Yeah, of course.
If you have problems accessing Sophie's email, most of you will know me and how to get in contact with me. So just email me and I'll put you in touch. So I'd like to bring this session to a close. Thanks, Sophie, again. I'm sure we'll be hearing more from Sophie and more presentations from the work you're doing. But thank you, and hope you, for the audience, I hope you have a good rest of the expo. So go and have a great start. But thank you. Thanks, Sophie.